So in these maple leaf mussels, which were the most common ones we collected at this site in the river, there's a good number here. You can see morphologically they're not all the same. This species does not have sexual dimorphism, so it's not a male-female thing. There is some age difference in there, which might account for some of the size difference, but they're not all the same regardless. So if you were to look at their bumps, various other features, they're obviously not all identical. So there's morphological variation within that species. But if we're interested in where they've come from, how they invaded the Great Lakes after the last glaciation period, morphological variation is not going to do it for us, is it?
No, it's something that's very difficult to quantify. So we have to look at, we have to take a small tissue sample from each of these, and then we can amplify certain parts of their genome. Usually what we call junk DNA. We use microsatellite markers that are developed for a very closely related mussel from the same genus. And what we can then do is, because these markers are highly variable, you can look at the genetic variation as something that we can easily quantify, and we can look at that at different site levels, at different watershed levels, and kind of across drainage basins.
So this is essentially a DNA fingerprinting kind of analysis.
It's exactly
very
the
similar
same
to what we did.
for DNA fingerprinting.
That's right. It's microsatellites for DNA fingerprinting in humans, for forensic work, for crime scene investigation. Same technology is being used here to study genetic diversity in these mussels and to understand their history, their movement from one region to another as the climate changed, and their overall genetic diversity in an evolutionary context. So it's very much the kind of thing you'd see on CSI, but for mussels.
CSI for mussels, eh? Call it that sometimes?
CSI for mussels.